Hi everyone! Today we're going to be walking through Chapter 3, which in this particular lecture we're going to be talking about another type of cell division, and this type of cell division is meiosis. So thankfully there are a lot of things that meiosis and mitosis have in common. So if you understood the process of mitosis, it won't be that difficult for you to understand meiosis as well. So we're going to talk about what meiosis is, how it's different than mitosis. Then we're going to talk about the formation of specific sperm cells and specific egg cells before our lecture review. All right, so two types of cell division that cells can undergo. Mitosis and meiosis. This, of course, assumes that we're talking still about multicellular organisms, right? So if you're a single-celled organism, you're really just going through mitosis. But as long as you are a multicellular organism, you have two options when it comes to cell division, mitosis and meiosis. Meiosis is a very specialized type of cell division, however. In meiosis, all we're doing is we are producing our sex cells. Those sex cells can be found inside gonads or sex organs. So the male gonads are testes and the female gonads are ovaries. As long as we're talking about animals in this case, plants are slightly different. We still do have male gametes and female gametes, but we don't refer to them as sperm and eggs. It's just a little bit different terminology. So these sex cells that we are producing through the process of mitosis are also referred to as gametes, male gametes, female gametes. Male gametes, again, in animals are typically called sperm, and female gametes in and females are, again, typically called egg cells. So when we're saying sperm cells and egg cells, we can also call those, again, our sex cells, our gametes. Now, what's interesting about our gametes and our sex cells is, remember, in order for a fertilization event to occur, in order to create a new organism, we need a full set of chromosomes. And remember, in humans, for us, a full set of chromosomes means having 46 chromosomes. So then we need to have sex cells that only contain half that information. We only need 23 chromosomes in egg cells and 23 chromosomes in our sperm cells as well. So the whole process of meiosis helps us to reduce the number of chromosomes by 1/2. So that way, when we have a fertilization event, the male gamete and the female gamete each have 23 chromosomes coming together to create those 46 chromosomes, the total number that we need in order to create a fully functioning human. So how do we decide which 23 chromosomes end up in each gamete? So we're starting off with a parent cell in meiosis that has 46 chromosomes. So how do we decide which chromosome goes where? Well, what we do is we do one of each kind. So if you remember, we had those 22 pairs of autosomes. Those are the chromosomes that we all have in common, both males and females. And then we had that one pair of sex chromosomes that determines our gender. So what we want is we want one of each of those kinds. So what we're going to do is we're going to take our chromosomes then, and we're going to separate them. So we're going to make sure that we have one set of autosomes in each of our sex cell, and then one sex chromosome in each of our sex cells as well. So we're going to be using kind of a new term today, talking about chromosomes and chromosome pairs. We're going to use this term called homologous pairs. So homologous pairs are pairs of chromosomes, one that you inherited from your mom and one that you inherited from your dad, and they contain essentially the same gene. Okay, they might contain slightly different versions of a gene, alternate forms of a gene, which we're going to call alleles in a future lecture, but basically they contain the same genetic information. So those 22 pairs of autosomes that I mentioned, they're numbered from 1 to 22. Number 1 is the largest chromosome, number 22 is the smallest chromosome. So your chromosome number 1 that you got from mom and your chromosome number 1 that you got from dad create a homologous pair of chromosomes. Okay, so they travel around in pairs with each other. Meiosis is going to split those homologous pairs up from each other. That's what we're going to do. So remember, in mitosis we talked about sister chromatids and separating sister chromatids. In meiosis we're going to talk about homologous pairs of chromosomes and separating homologous pairs. A non-homologous pair of chromosomes would be two different chromosome numbers. So chromosome number 4 from mom and chromosome number 1 from dad would be a non-homologous pair. They have different genes, different genetic material. They don't pair up with each other naturally. So in meiosis, we're going to separate the homologous pairs of chromosomes so that each cell only has one of each pair. And remember, this is a specific term called having a haploid number of chromosomes. So we call these cells haploid cells. Haploid just means half, right? Haploid, half. We have half the total amount of genetic material that we need. We don't need 46 chromosomes in our egg and sperm cells. We only need 23. So we're going to cut our genetic information in half. And again, depending on whether we're talking about males or females, meiosis is occurring in either the testes or ovaries. So testes for males, ovaries for females. And again, once we combine the eggs and the sperm cells, we would get a zygote or a fertilized egg. The fertilized eggs then are diploid because, again, we had an egg with 23 and we had a sperm with 23. They came together. They created a diploid cell. All right, so diploid is two, having two of each kind of chromosome. So that's the whole purpose of what we're doing here in meiosis, is we're making it so that when we go through the process of sexual reproduction, we have the right number of chromosomes. All right, so let's talk about some things that we've already discussed before. It'll be a little bit of review for you. Just like we had in mitosis, meiosis is preceded by interphase and then followed by our division phase. So just like we saw in mitosis, we have the two gap phases and we have a synthesis phase. So we are copying our DNA, we're getting all of our cellular structures in place that we need to go through meiosis. So interphase in meiosis is just like interphase in mitosis. In our cell division, we have, again, similar terminology. The only difference is that we have to divide twice in meiosis. There's a meiosis 1 and a meiosis 2. In meiosis 1, we're going to separate the homologous pairs of chromosomes from each other. In meiosis 2, we're going to separate the sister chromatids from each other. So for all intents and purposes, meiosis 2 is identical to mitosis. Okay, so meiosis 2, identical to mitosis. Meiosis 1 is what's a little bit new, and we'll talk about that more. So here's a nice overview of the process. We're going to start with a diploid cell. It means it has two copies of each chromosome. Meiosis 1 is going to reduce that, so we only have one copy of each chromosome. So at this point, we'll be a haploid cell. And then we take apart these sister chromatids, because remember, sister chromatids are just replicated genetic information, and we're going to separate those out. So another key difference here is that in meiosis, we end up with 4 daughter cells rather than 2 daughter cells. All right, so let's talk about meiosis 1. We get to use the same phases as we did in mitosis. Yay! So it's prophase, metaphase, anaphase, and telophase, just like we saw before. Except now we say prophase 1, anaphase 1, telophase 1, whatever it happens to be, because we're talking about meiosis 1. So a lot of things we've already seen before. In prophase 1, we have homologous pairs coming together, condensing, the nuclear envelope disappearing, lots of stuff we've seen before. In metaphase 1, the homologous pairs line up at the equator. In anaphase, they separate, and in telophase, nuclear envelope reforms. So again, we've seen this. So let's take a look diagrammatically at what this looks like. Again, this should look very, very similar to mitosis because you've seen something like this before. So our centrioles and our spindle fibers forming, our nuclear envelope is going to start disintegrating in prophase. We're going to get our homologous chromosomes to do something unique called synapsis and crossing over, which I'm going to talk about in a moment. I'm not going to talk through all of that right now, but I'm going to talk about it in a moment. In metaphase, we're going to have those line up at the equator, and then we're going to separate those homologous chromosomes in anaphase, and then we're going to have nuclear envelope reforming in telophase, and then we have a cytokinesis event as well. Oh, I'm sorry, I just realized that you guys can't see. Here we go. There we go. So prophase, synapsis, and crossing over. Prophase 1 late, condense, they become visible, spindle forms, all of those things that we've talked about. In metaphase, they line up at the equator. In anaphase, the homologous chromosomes separate. In telophase, we put the nuclear envelope back around, and cytokinesis divides the cell in two. Okay, so again, the big difference here, I'll talk about crossing over, and I'm also going to talk about something called independent assortment, but that's a good overview for meiosis 1. Meiosis 2, hey, look, same thing, prophase, metaphase, anaphase, telophase. This time, we're taking those two cells that just came out of meiosis 1, and we're going to put those through meiosis 2. And again, for all intents and purposes, meiosis 2 is identical to mitosis. Same thing happens, okay? Same thing happens in prophase with the nuclear envelope disappearing, and we have in metaphase those sister chromatids, right? Because we're still attached to that duplicated material lining up in the equator. We separate those in anaphase 2. Telophase 2, we create those nuclear envelopes around our genetic material again, and then after cytokinesis, we end up with 4 haploid cells. Haploid meaning, again, they have half as much genetic information. The other key here is that every single one of these cells are different. So in mitosis, our whole goal was to create identical copies of cells, just to replace the cells as we needed them. Meiosis has a different goal. We don't want identical cells. We want genetic variation. We want as many genetic combinations as possible. It's why you can have 2 individuals who can have as many children as they want, and as long as they don't have identical twins, not a single one of their children will be the same, because each child is a new combination of chromosomes that came from the sperm and chromosomes that came from the egg, and it creates a new combination of traits, and that's what we want through meiosis. So again, prophase, metaphase, anaphase, telophase. Okay, as far as meiosis 2 is concerned, it's just like mitosis, separating sister chromatids. So again, since there are 2 separations, we end up with 4 cells. Oops, sorry, let me adjust this again. Okay, quick comprehension check. See if you guys know this. We've got a diploid mouse cell that has 40 chromosomes. When a diploid mouse cell goes through meiosis, there are blank chromosomes in each daughter cell. Okay, so let's say that mouse cell with 40 chromosomes, it went through meiosis. Each of those 4 daughter cells it creates is going to have how many chromosomes? Will it have 40, 20, 80, or 10? It's half as many, right? So we're going to go down to 20. 40 would be the same, so that's not it. 20 is half, that's exactly what we're looking for. 80 would be double the number. We don't need double the number. That's not necessary. We need that perfect half as many chromosomes in each of those daughter cells. All right, let's talk about genetic variation. So, all of these cells that we're making are different genetically. And again, it's why siblings from the same parents produce genetically different offspring, with the exception of identical twins who have the same chromosomes. So that's why they're identical. This is important because, again, we're creating genetic recombination and variability. Why is this important? Well, it's important because we don't all want to look the same. We don't want to all have the same traits and the same genes. It's necessary because if something does happen, if a new virus comes in, a new flu virus comes in, and this happens often, some people are really susceptible to these flu viruses and they can be deadly, and other people aren't susceptible at all and they won't even get sick. That's part of that genetic variation. So we need to have that in a population of living things so that we can be able to survive our environment. So the first step in creating this genetic variation is something called crossing over. So when the homologous pairs are together during prophase 1, they do something really interesting. They actually swap genetic information. So let me show you this diagram here real quick. And again, let me adjust this so you guys can see my screen. So here's our homologous pairs of chromosomes, okay? So let's say dad's is blue and mom's is green, okay? Here are the different genes on our chromosomes. What happens is what's called a synapsis event, where these chromosome arms are going to kind of hug each other a little bit, and they're literally going to switch pieces. So a little bit of dad's chromosome is now on mom's chromosome, and a little bit of mom's chromosome is now on dad's chromosome. So there are an endless number of combinations that we can create by doing this event called crossing over. So if you didn't have crossing over occurring, okay, let's say it didn't occur, and here we have some plants that we're talking about, and the 2 genes we're looking at are flower color and the type of rice grains that we have, okay? So in this case, we have red flowers and we have white flowers. and we have long grains of rice and short grains of rice. So if crossing over didn't happen, at the end of meiosis, this is what we would end up with. We would only end up with two types of gametes, right? We would have gametes that were red with long grains and gametes that are white with short grains, which is kind of boring. But when crossing over does occur and we swap a little bit of genetic information, now we have four types of gametes. So we still have the red long and the white short, but now we also have a red short and a white long. So again, increasing genetic variability. That's what it's all about. Next idea to create genetic variability is something called independent assortment. An independent assortment simply means that when your chromosomes are lining up during metaphase, not all of the same chromosomes line up on the same side of the equator. So it's not like all of mom's chromosomes line up on the top of the equator and all of dad's chromosomes line up on the bottom of the equator, because again, that wouldn't create a lot of genetic variability. Instead, what we get is we get a flip-flop of how they're lining up, and so it creates again a new combination of chromosomes and traits. So again, just sort of diagrammatically, if we have them, dads and moms are lining up on the same side, we don't get a lot of variation. However, when we flip those and we switch the order in which they're lining up, we get new combinations of chromosomes. All right, I apologize, I have to continue to change my viewing screen here. Comprehension check. Which of the following statements about meiosis is false? Daughter cells are genetically different than the parent cells. Meiosis occurs in germline cells. DNA replication occurs twice. Crossing over occurs between two homologous chromosomes. This one's a little tricky. It's actually number 3. DNA replication occurs twice. There's only one replication event here, and that happens in interphase before meiosis 1. Okay, that's it. That's the only time we replicate our DNA. We don't need to do it twice, because remember, we're trying to get half as much genetic information, not double the amount of genetic information. Okay, so this is a really nice table from your textbook that actually goes through and compares the similarities and differences between mitosis and meiosis. So the number of divisions, how many daughter cells we get, whether they're identical or not, whether they're haploid or diploid, where it occurs. So this is a really good way to remember the differences between mitosis and meiosis. I highly recommend studying this chart here. Last thing I'm going to briefly mention is sperm formation and egg formation. So we call it spermatogenesis. You can essentially see that there's a cell called a spermatogonium, and it produces what are called spermatocytes. And then these spermatocytes will divide over and over and over and over to continually make sperm. So as soon as males hit puberty, they will continue to produce these sperm cells essentially until the day they die. Oogenesis is a little different. So in oogenesis, we have again an oogonium which produces these primary oocytes, and these primary oocytes will then divide and go through the process of meiosis, creating eggs. And as you can see, it's not exactly equal. So in spermatogenesis, we had all these sperm cells constantly being created. In oogenesis, we go through this process and we only get one of these eggs. So all of these other cells tend to be what are called polar bodies, and they end up dying off. And all we're left with is this one cell out of the process that's a mature egg. The other thing that's very different is when this occurs. So basically, oogenesis and creating these primary oocytes happens before ladies are even born. So it's happening when they're in the womb. This is all happening. And then during puberty, the primary oocytes will then again produce what are called these eggs. But you're going to eventually run out of eggs. So once you run out of the primary oocytes, that's it, ladies. There's no more eggs after that. Whereas males, they're born with a spermatogonium, which will then produce more spermatocytes, and then they'll continue the process again, essentially until the day they die. All right, here's our lecture review, important terms and concepts to know.